Welcome to the third lecture in our lecture series in this course. In the previous lecture, we looked at algae and we said algae are thallophytes. Now today, we are looking at fungi, and fungi is the second group in the lower plants we call cryptogams. Now fungi can be described as eukaryotic microorganisms which are heterotrophic and are essentially aerobic with very limited anaerobic capabilities. That means they can only survive in an area that has oxygen. Fungi, if you look at their mode of nutrition, you are saying it's heterotrophic, you realize that they normally digest their food externally by releasing hydrolytic enzymes to the immediate surrounding. And that's why you'll find fungi on dead, decaying wood, like in this case here. This and this are edible, and even this one here is already packaged for eating. There's somebody called Pasteur. Pasteur said that without fungi, the process of death would be incomplete. After materials have died, they need to decay and be taken back to the substratum. So what fungi does is that it breaks down all the organic matter that had been absorbed by a living organism, and then this matter is taken back to the soil in a process we refer to as mineralization. I've already said what fungi is, and therefore let us now move to the classification of fungi briefly. In the previous lecture, we said that the plant division names always end in phyta. Now for fungi, there is a difference. Division names in fungi end in mycota. M-Y-C-O-T-A, mycota. Class names, we said, will always end in opsida. But for the case of fungi, the class names end in mycetes. So now, when you classify fungi into classes, the 5 broad classes that you'll find almost all classification systems referring to are: number 1, the Mycomycetes. These are the slime fungi. We have the Phycomycetes. These are the alga-like fungi. We have the Ascomycetes, which are the sac fungi. We have Basidiomycetes, which are the club fungi. And we have the Deuteromycetes, which are the fungi imperfecti. The Deuteromycetes are referred to as the fungi imperfecti because in their life cycle they do not exhibit any sexual phase. The general characteristics of fungi. Fungi are a group of thallophytes. That means their bodies are thallus and they do lack chlorophyll but have other pigments in their cell wall. Remember, in algae we had chlorophyll. They have a variety and an infinite variety of shapes and sizes. Of course, their mode of life is heterotrophic, but you'll find some living as either saprophytes or as parasites. The carbohydrates, the reserve carbohydrates in fungi is in the form of glycogen. You remember we mentioned this when we were looking at algae. We talked of starch in algae. Now, the fungal parasites are either obligate or facultative. Obligate in the sense that their entire life depends on a host. When you remove a host, then the fungi cannot survive on its own. But if it's facultative, it means that at a given time in the life of the fungi, it can survive in the absence of a host. Now, fungi can also survive and live on the host as a saprophyte. And a good example are smats, which we'll be seeing towards the end of this lesson or lecture. Now, the plant body of fungi is made of an interwoven mass of hyphae, which are collectively referred to as a mycelium, and there are very few exceptions. So here we are having a body of the fungi being called a mycelium. If you look at the wall of the fungi, you realize that it is made of chitin or prune cellulose. Reproduction. Now, anytime we are talking about reproduction, to know that reproduction can either be vegetative, that is when portions of the plant body are used to generate a new being. It can be a sexuating on a suitable substratum. When we can also have sexual reproduction, and in this case we shall have the male and female gametes uniting through fertilization and resulting into the development of a new being when a zygote is formed. So vegetative reproduction in fungi can either be via fragmentation, where a portion of the body breaks off and develops into a new organism, a new fungi. You can have a portion of the fungi detaching from the main body, and then you can also have, in some cases, just a budding on the portion of the fungal or fungal body. Vegetative reproduction can also be through special bodies referred to as sclerotium. And what is sclerotium? Sclerotium is a compact and is often a hard, rounded mass of hyphae that's occasionally with a darker outer fruit covering without any spore. And these are of different sizes. So this sclerotium will always give rise to a mycelium or a fruit body. Asexual reproduction. This one takes place by means of spores of varying nature, and they include the ciliate spores or zoospores. Ciliate spores are spores that have cilia. Anytime you hear of the term zoospores, it will mean that these are spores that are motile. Asexual reproduction can also be via ordinary spores, which are produced in large numbers in a casing that is known as the sporangium. Sometimes conidia are also formed, and these conidia are the ones that will be released from the fungal body, and then they germinate just like spores. So these conidia can either be formed singly or they can be produced in chains in a specialized hyphae or a conidiophore at the tip by abstrictions. Abstrictions are just breakages or breakings. So when you are talking about conidia, this is what you are referring to. This portion here, these are single conidia. It detaches. This is a series of them, and this is a conidiophore. Asexual reproduction can also be via chlamydospores. And chlamydospores are thick-walled resting spores which are formed singly or in chains by certain vegetative hyphae. And these are always normally transformed to a vegetative cell. We also have oidia, and oidia are just short segments of a vegetative hyphae that are functioning as spores. We can also have ascospores, and if you hear the word asco, it means it's from Ascomycetes. So ascospores, these are mostly produced as 8 in number, and they are formed within a sac or an ascus. We have said that they are normally 8 in number. If you count this, you realize 1, 2, 3, 4, 5, 6, 7, 8. So this is the ascus that is holding the ascospores. We also have basidiospores, and basidiospores are normally 4 in number, and these ones are formed externally by a club-shaped basidium or a short slender stalk called sterigmata. And this is what we are talking about. These are basidiospores. If you count, this is 1, 2, 3, 4 of them, and we are saying that they are held by a slender stalk called sterigmata. This is the sterigmata. So anytime you are in the laboratory and you are observing spores under a microscope and you see 8 of them being produced by the ascus, then this will be an ascospore. If you see 4 of them, then that should be a basidiospore. Sexual reproduction. Now in sexual reproduction, this involves the union of gametes, the male and the female. Now this process of sexual reproduction goes through distinct phases. First of all, what happens is the process referred to as plasmogamy. Now plasmogamy is the fusion of 2 protoplasts without the fusion of the 2 nuclei. So you'll have a protoplast moving and getting closer to another one, and then these 2 protoplasts will fuse. But as they fuse, the nucleus that are within them, the 2 nuclei, do not fuse. Now, the 2 nuclei of the opposite sexes that have been brought together in these protoplasts that have fused constitute what is called a dikaryon. That cell that has now the 2 nucleus, but the nuclei that have not fused, is a dikaryon. And this condition is what is referred to as a dikaryotic condition. So now, the dikaryotic condition may persist for some time, like in the case of the higher fungi, where the 2 nuclei, but in the case of the lower fungi, the 2 nuclei will fuse immediately. Now, at that point where the 2 nucleus within the dikaryon are fusing, that time they constitute the process of karyogamy. So karyogamy is the fusion of the 2 nuclei. Anytime you are seeing a positive and a negative, positive denotes male while negative denotes female. So the 2 nuclei of the dikaryon will fuse, resulting into a diploid zygote nucleus. So what happens is that meiosis follows, and later on it reverts into a haploid condition. Now, with the development of gametangia, sexual reproduction may be isogamous or anisogamous or oogamous. Now, isogamous refers to a situation where the male and the female gametes are similar in morphology, and the size and the shape are also the same. In anisogamous, the male and female gametes are not similar in morphology, so it means that they vary in the size and even in the shape. In oogamous, oogamous is a form of anisogamous where the female gamete is significantly larger than the male gamete. Characteristics of the main classes. We'll start with the Phycomycetes, the alga-like fungi. Now in Ascomycetes we have the oomycetes and the zygomycetes. In the oomycetes are biflagellate. Their sexual reproduction is oogamous, and an example is Pythium, Phytophthora, and Peronospora. The second category of Phycomycetes are the zygomycetes, and now reproduction in them is isogamous. Here the male and the female gametes are the same, the morphology is the same, and they have also a similar shape and size. A good example is the Muca and the Rhizopus. Now in zygomycetes you'll find that the mycelium is uniseptate, and you can also say it is cynocytic. Now when you're talking about a uniseptate mycelium, these are mycelium that has no septa within it. There are no divisions like in this one here. It is entire nucleus. The nucleus is spread, but there are no divisions within the mycelia. These are cynocytic hyphae. It can also be said to be aseptate. Now the sporangia has so many sporangiospores, which may be zoospores or aplanospores. Now the sporangiospores we are saying can be either zoospores or aplanospores. Zoospores are those spores that are motile, while aplanospores are non-motile spores. Now the zoospores are normally biciliate in many species. The zygote is unicellular and it is normally simple. Now we talked of the mycelium being uniseptate or cynocytic, but in the event that the mycelium has, that is in other fungi, not in Phycomycetes, I'm just using this diagram to explain this. When you have septa, then this septum, this mycelium, septum within the hyphae is now referred to as being septate. Now sporangiospores are produced from sporangia. Sporangia is just the plural of sporangium. And this diagram here, this is sporangium. So the spores that are found in this sporangia are sporangiospores, and the ones we are saying can either be zoospores or aplanospores. Ascomycetes. Ascomycetes are the sac fungi. Their mycelium is septate, the way I've shown you in the previous slide, and there are two types of the septate mycelium. We can either have the primary mycelium, which is normally uninucleate or multinucleate, or you can have a secondary mycelium, which is always binucleate. Now, in the asexual stage of the Ascomycetes, you'll find that conidia formation is very common. Now, the ascus usually has 8 ascospores. Sexual reproduction in the Ascomycetes is just reduced to the fusion of a compatible male and female nuclei, and that's karyogamy. Now, once karyogamy has occurred, it is immediately followed by meiosis, and this results in the 8 ascospores within the ascus. In the Ascomycetes, we do not have motile cells, so motile cells are absent. The ascoscarp is usually the fruiting body, and it bears the asci. It is multicellular and complex. When you look at this fruiting body we are referring to as the ascocarp, then there are three types. Three types of ascocarps can occur in the Ascomycetes. We can have an ascocarp that is referred to as an apothecium. This one, it's open and it's either cup or saucer shaped. And this is the diagram. It's open, you see it's saucer shaped or it's cup shaped. This is an apothecium. An ascocarp can also be said to be a perithecium, and in this case it's normally oval and flask shaped with a small apical opening. This is a perithecium. Sometimes the ascocarp is completely closed, and that will be referred to as a cleistothecium. Basidiomycetes. Basidiomycetes are the club fungi. When you look at the mycelium of the club fungi, it is septate, which means that's a septum. Now, the primary mycelium is multinucleate, while the secondary mycelium is binucleate. You will not find conidia easily, so conidia is not common in the Basidiomycetes. Now, the basidium usually has 4 basidiospores, which are formed exogenously. Again, in this group of fungi, sexual reproduction is reduced to the fusion of the 2 compatible nuclei in the young basidium. Now, there is no sexual organ except spermatia, which are receptive hyphae and are receptive hyphae in rusts. Karyogamy is immediately followed by meiosis, resulting in 4 basidiospores, which are born exogenously by the basidium. Again, in the Basidiomycetes, motile cells are absent. Now, the fruiting body in the Basidiomycetes is called the basidiocarp. It's normally multicellular, complex, and it bears basidia. Now, the basidiocarp is often open, but sometimes it can be closed. Lower members in the Basidiomycetes, like the rusts and smuts, have no basidiocarp, and you'll find that clamp connection is very common in this group of fungi we are referring to as the Basidiomycetes. We want to look at an image that will help us understand what a rust is and what a smut is, because we have said that these are lower members in this category. If you have been careful at home, you must have realized that when you are harvesting maize or fresh, before it dries, you will realize that it looks something like this. This is just smut, and this is specifically Ustilagomycetes. Now, rusts appear like brown patches on the leaves of plants. In this case, we are having Puccinia hordeae. This is brown rust. The economic importance of fungi. Fungi can either be useful or harmful to human. Now, in the field of medicine, There's an antibiotic which was discovered and is called penicillin. Penicillin was obtained from a fungus called Penicillium notatum, and this became a wonder drug in the 1940s for the treatment of bacterial diseases. It gave fungi a niche in the science of the discovery of antibiotics, and today several important antibiotics are produced by molds. Now, many fungi are edible, like the yeast, the mushrooms, the truffles, the morels are all edible, and this is food for human. Now, edible mushrooms are known to contain proteins and vitamins. Certain species of agaricus, such as Agaricus bisporus, Agaricus arvensis, are edible. And even species of Volvariella like Volvariella volvacea and Volvariella dispora are also edible mushrooms which are cultivated commercially. Now, the brewing and baking industry relies heavily on the use of yeast. This is Saccharomyces. The yeast always, we know, ferment sugar, and the sugar solution that is fermented produces alcohol and carbon dioxide. The alcohol is the one that is used in the brewing industry, while carbon dioxide is used in the baking industry. I've already said that minus fungi, Pasteur said that without fungi, even death will be incomplete. Now, what fungi ensures is that the dead cellulosic vegetation is decomposed into carbon and minerals by saprophytic fungi, and these elements are returned to the same environment where they were obtained. So fungi are very important in maintaining the carbon and mineral cycles in nature. Harmful aspects of fungi. Now, fungi are a great nuisance. Anything that is moist, you realize that they'll be growing on jam, on leather, and they'll be spoiling anything in the house because the spores are always in the air and they germinate easily. Now we have LSD. LSD is D-lysergic acid diethylamide. This is normally a hallucinogen that is produced from the fungus ergot. Now, this is a substance that is psychoactive and affects human. Common fungal diseases of plants are like the wilt of cotton, which is from Fusarium oxysporum. We have the ticker disease, or the leaf spot of the groundnut, that is as a result of the fungi Sarcospora personata. We have the red rot of sugarcane, which is as a result of the fungi Trichum falsatum, Coletotrichum falsatum. Now, fungal diseases of human beings include like the ringworms. This is an Epidomophyton species. That's the genus Tinea. We have the Trichophyton species also causing ringworms. We have Candidiasis, and this one is just Candida albicans. Claviceps purpurea also produces hallucinogens, and these hallucinogens are the ones responsible for hallucinations, and that's why they are referred to as the hallucinogenic fungi. They cause a great damage to the frustrated youths because these are used like drugs. They are used like drugs, and they give the youths a feeling of an unreal, extraordinary lightness that gives them a hovering sensation. What they do is they are very psychoactive, and they give you hallucinations. These are examples of fungi that are harmful to human. This athlete's foot. You can see what's happened to this person's legs. This is a ringworm. And this is the red rot of sugarcane. Sometimes when sugarcane is kept for long, you find it turning to red. This is just a fungi. Thank you. This is the end of fungi. Next, we shall be looking at lichens.